From counting birds at Christmas to cataloging bacteria in the human microbiome, citizen science is a template of scientific participation that can be used by teachers to engage large numbers of students in research. Assisting with lab classes is one of the best opportunities for early career scientists to start teaching, but designing these classes requires a different skill set altogether. How can you get ahead of the game and leverage citizen science to synergize your research with your teaching? If it's your first time here, my name is Jack Wayne, a microbiologist, science educator, and a 2020 Australian University Teacher of the Year. In this new series of videos, we are talking about lab techniques and research questions that can be adapted for course-based undergraduate research experiences or cures. You'd be surprised how few science students actually get involved in research throughout their studies. It's less than 10%. And cures embed semester-long research projects as part of the standard undergraduate experience. Seeing as cures provide a mechanism for large-scale student participation in research, the natural progression gaining momentum over the past few years is citizen science, where research participation is further extended to the general public. We will outline the connections between cures and citizen science and describe how both can provide meaningful ways to better integrate your research into your teaching and synergize professional development as an early career academic. In the last video, we talked about recombinant DNA technology and how molecular cloning of specific genes can be adapted for citizen science projects. Following on from cloning novel plasmids that encode genes of interest, students can work with their own plasmids or characterize existing plasmid libraries generated by your research labs to express and purify novel proteins of interest. Just like molecular cloning, in protein expression experiments, if one step fails, everything fails. Giving students more autonomy in experimental design does not have to be impossibly difficult. You can limit the variables that students can change in each experiment and provide a scaffolded, inquiry-driven learning activity. It helps that each protocol in protein chemistry has many variables that need to be optimized, including incubation times, reagent concentrations, and choice of buffer solutions. Students can be given a high degree of autonomy to survey the literature in search of the best experimental conditions for their protein of interest. Students can be allocated a protein of interest to work on or choose from a subset of closely related novel proteins. The experimental workflow involves transformation of plasmids into cells, most commonly using bacterial expression systems, induction of protein overexpression, and detecting of protein levels through STS Page and Western blot. We have videos covering quite a few protein expression techniques on this channel, all of which are linked below. After this point, it will depend on the features of the recombinant plasmid and the function of the gene product of interest. You may ask the students to purify the protein using a variety of different techniques, gel filtration, ion exchange, or affinity chromatography, and or run functional experiments to measure protein activity. Of course, there are almost an unlimited number of ways of defining protein activity, but focusing on proteins with known enzymatic activity is a pragmatic approach. You can run colorimetric assays with the enzyme activity results in a color change, which can be read by a plate reader, and very quickly analyze different conditions with many different protein products. As always, I recommend that you consult the science education literature. There are many more published examples of cures than you might think. My go-to for protein chemistry research projects is BAMBED, which is many years worth of peer-reviewed lab exercises that focus on measuring enzyme kinetics of different proteins, Taq polymerase, malate dehydrogenase, and different phosphatases. And in each case, the substrate and products are well known. PCR and spectrophotometric assays are standard operating protocols in these types of lab classes, so you can feel free to give students more creative freedom to change the experimental variable. This mode of inquiry neatly lines up with site-directed mutagenesis experiments, systematically mutating individual protein residues one by one to see how and when the protein function changes in response to these mutations. These mutant plasmid libraries are very useful to research labs, but are otherwise considered a routine mechanistic exercise for researchers. So empowering undergraduate students to do this work at scale makes sense for everyone. Remember the three components of citizen science: authenticity, scalability, and communicability. And this work is already authentic to the work researchers do in their labs. Students can either be involved in generating the mutant plasmid libraries or be given an existing mutant plasmid library to work with. There could potentially be hundreds of mutants for one single protein, so even in a class with hundreds of students, you can give each student or pairs of students their own mutant to work on. The mutant protein is then expressed, purified, and its enzyme activity measured. And the data across the whole class is pooled to figure out which amino acid acid residues are directly involved in the enzyme activity of the wild-type protein. Hopefully you can see by now that the iterative nature of these protocols needing to be repeated for each mutant inherently makes these lab activities scalable, a great fit for large class sizes and ticking the scalability box for citizen science. The last component is of course communicability. And just like molecular cloning, the hook has to be based on the function of the protein students are working on. Making the protein of interest immediately relevant to diseases and essential biological processes is what connects the work to people's everyday lives. And if we look to the scientific education literature, you can get a sense of what other people have done in the past. Cures and citizen science projects have investigated proteins implicated in diabetes, obesity, brain disorders, and a broad array of different cancers. Molecular cloning and protein expression techniques are universally useful in any biological project, so there is no reason why you can't ask students to work on a protein of interest in your own research. By forcing yourself to communicate why your protein should be interesting to the students, this gives you more reps and communicating the importance of your work to different audiences, which of course all ties in with your professional development in both research and teaching as an early career scientist. That's it for now, but this isn't the last video in this series on cures and citizen science. This is the Biolab Collective. I'm Jack Wayne, and I'll see you in the next video.